Using social media, we're using the hashtag REGEN2015, and those of you watching online can feel free to submit questions to our panelists using that hashtag.
Okay, so can everybody hear me okay? So I want to try to encourage you guys to raise your hands, ask questions. We've got a great panel here. And what I thought we'd do today is I'm going to have our panel introduce themselves, where they're from, and perhaps their area of expertise. And then I'm going to start out with a series of questions to get the conversation going. I would encourage follow-up questions. This is going to work best if you hear less of me, okay? So, John, perhaps you can get started.
So I'm John Epstein. I'm at the University of Pennsylvania. I'm trained as an adult cardiologist, but do mostly basic science research. I've been chair of the Cell and Developmental Biology Department there for a number of years, and I'm the chief scientific officer there. And many of you heard the work we do today. I'm mostly interested in the development of the heart and vasculature and molecular pathways that regulate stem cell and quiescence and regeneration.
Hello, I'm Rick Morimoto. I'm a professor of molecular biosciences. I do biochemistry and neuroscience at Northwestern University in Evanston, Illinois. My area of research is really to try to understand questions of why do we age, when do we age, how do we age, because yes, well, when it doesn't go as perfectly as you imagined, one becomes at risk for different diseases. And we're trying to understand quality control mechanisms, how to make the cells and tissues more robust so that we can resist the temptation for failure.
Okay, I'm Eric Olson. I'm from the University of Texas Southwestern Medical Center at Dallas. I'm the chair of the Department of Molecular Biology, and have also founded a Center for Regenerative Science and Medicine in Dallas. My lab works on muscle, both heart and skeletal muscle, and we've focused on that problem for 30 years. We're interested in deciphering the basic mechanisms that form muscles and understanding how those basic mechanisms can be leveraged to regenerate and repair damaged muscles during a number of diseases. In particular, right now, one of the ones that we're extremely enthusiastic about working on is muscular dystrophy, which, as you know, is a devastating disorder of skeletal muscle.
So I'm Tom Rando. I'm trained as a clinical neurologist, and that's my day job is to run a neurology service actually at the Palo Alto VA Medical Center. I'm a professor of neurology at Stanford University, and my lab has been interested in also skeletal muscle and muscle regeneration, muscle diseases, and we've been very focused on stem cell biology recently and tissue repair, and then also this issue of declining regenerative potential with age, trying to understand regulators of stem cell function across the age spectrum to enhance tissue repair in older individuals. Also, another interest in the lab, and I run a regenerative rehabilitation center at the Palo Alto VA, which is sort of an intersection between regenerative medicine and rehabilitation medicine. And we have a growing interest in using tissue engineering to reconstruct tissues using a combination of stem cell biology and primarily material sciences, so a very kind of translational aspect as well.
I'm Nadia Rosenthal. I'm the only one you haven't heard from yet. I am currently in transit. I am the director of the Australian Regenerative Medicine Institute at Monash University in Australia. And that day job is going to segue into being the scientific director here at the Jackson Laboratory a couple of miles down the road. And in the middle, I spend part of my time in London as a chair in cardiovascular science at Imperial College London. So my research is, like many of the others on the panel, focused on the basic science behind regeneration, and I've become more interested recently in looking at various models, animal models of regenerative potential, of which we've heard quite a few today. Specifically, we've become very invested in axolotl biology to try to use that incredible model of perfect regeneration as a way to understand why we don't do such a good job at regrowing our limbs and fixing our spinal cords. And more recently, in conjunction with my new job at the Jackson Laboratory, I've become very interested in the question of what is the regenerative potential variation within a species? How is it that some people can regenerate much better than others, can recover from a heart attack much better than others? And my focus on the heart and skeletal muscle has really now branched into this question of a more general capacity for regeneration and looking at the genetics of those processes.
Great. So the first question I'm going to throw out there is obviously the one that we're all thinking about, and that is there's been a lot of hype. There's been a lot of hope in terms of what regenerative medicine can deliver and the potential. But there's also been a lot of controversy, what's gone right, what's gone wrong. So I'd like to hear your perspective on what you think some of the key barriers are to making a lot of the potential of regenerative medicine becoming a reality. And then the second part of the question is, what particular aspect of regenerative medicine do you think would be the best area for success within the next, let's say, 10 years?
I think it's important that the general public realize that regenerative medicine is more than just stem cells. I think stem cells have been, it's my own personal opinion, been overhyped. In the literature, there's been a push to deliver stem cells for a variety of therapeutic indications without a deep understanding of the underlying biology, sort of a philosophy of, well, if it works, who cares how it works? But the fact is, there have been many stem cell trials that haven't worked. With respect to, you mentioned the barriers. I think some of the barriers are unique to humans that are not seen in the types of model organisms that we've talked about at this meeting. So there's, as all of you know, there's some invertebrate organisms and simple organisms that have just amazing regenerative potential. We heard about some of those today. They can recreate themselves from just a piece of themselves. They can regrow limbs, regrow hearts, regrow organs. But one of the huge barriers with respect to doing those things in people is size and complexity and age. So size, if you think about regrowing a limb of a salamander, it might be the size of half of my pinky, but if you think about regrowing an arm of a human, that you've got a much bigger problem to deal with. I think complexity in terms of the cellular and genetic complexity is a barrier. And although not talked about so often, I think age can be a barrier. Our group showed several years ago that newborn mice can regenerate 20% of their hearts following surgical amputation or injury, whereas adult mammals cannot do that. And I think with respect to humans, the older a person gets, the more locked in their genome becomes. It becomes less plastic and more difficult to revert it back to a regenerative state. So I think those are some of the challenges that are posed by regeneration of large tissues and organs in humans.
Well, I'll just respond to your question, turning it on its head a little bit. I think regenerative therapies are very successful, and it's a spectrum, of course. We'd love to be able to do much more, but we often forget that we use bone marrow transplants regularly. It's a very successful regenerative therapy. Even wound healing approaches, transplanting skin, growing skin. And even the sort of routine medical therapies that we don't think of as regenerative therapies, like conservative treatments for acute renal failure, are really regenerative therapies that are allowing the kidney to regenerate its tubules on its own. And I think what we may find is that there's a lot more emphasis on tuning the body's own natural ability to regenerate, boosting it where we can. As opposed to the heavy emphasis on stem cells. I think The stem cells can be very good. I think there's a lot of hope for stem cell therapies. But I suspect that regenerative therapies will widen in their spectrum, small molecules being used to manipulate the natural regenerative processes. And we may not be able to declare a day when suddenly it works. It'll be a gradual process of small victories.
I think one of the problems with the stem cell field, which John and I think share our skepticism about much of what's being touted as a therapeutic direction, is that the approaches are quite Neanderthal. You grab something out of somebody's fat, whirl it around in a blender, and throw it back in with a needle and expect it to take. It's a bit like dropping someone with a parachute into the Gobi Desert without a compass or an iPhone, And I don't think they know what they're doing. That is to say, I don't think the cells have a clue what they should be doing when they get there. And one of the issues is that nobody really understands what it takes to tell a stem cell what to do. And the whole concept of the niche for what has been known, for instance, in the bone marrow as a very complicated and even now poorly understood interaction between the environment in which a cell grows up and its capacity to then manifest as a particular progenitor has really, we haven't gotten far enough to be able to understand what it takes. And mixing cell A and cell B is not what I'm talking about. I'm talking about actually knowing the biology of how a stem cell behaves and why it does what it does when it does normally and why it doesn't. I think that's one of the major reasons why one after another these stem cell trials are coming up not particularly successfully. So more basic science.
More basic science. There are questions in the audience. If you just raise your hand, we have microphones that will find you. In the meantime, it's sort of a segue from that. You mentioned the microenvironment and then the niche. And obviously this has been shown to be important in a number of contexts, skeletal muscle, heart, of course, limb regeneration, Nadia, with some of your studies. Do you think that there's going to be an underlying code or circulating factors that one could potentially identify that are going to have multiple effects regardless of the type of injuries and where the injury is located?
So I'm not sure I would predict that there are going to be the same factors in different tissues. I mean, I think getting back to something that Eric mentioned, back to the issue of age, I think that one of the challenges there, and this will come back to systemic factors, is that it's very likely, it seems to me, that one of the reasons why regeneration declines with age, when I say age, I don't just mean, from adulthood to getting older, I mean from birth onward, or even from development onward, is that there's almost certainly a built-in program for suppressing growth at a certain point when growth stops. And so this may be one of the trade-offs. It's almost like a systemic tumor suppressor biology that actually is a trade-off between suppressing cell growth and allowing cell growth. And the older we get, the more we would want to suppress growth of cells that may have genomic instability and other abnormalities. So that gets into this issue of there being within the niche a kind of biology that makes it, we're fighting against nature to get stem cells to do what they do developmentally in an adult. And then I would predict that, yes, that there are probably not only local factors within the niche, but the entire systemic environment is changing over time in a way that is more likely to suppress stem cell function. So whether it's small molecule induction of endogenous stem cell function or transplantation of stem cells, they're all fighting against that. And it may be possible to change those and to promote better growth, but at some risk. I mean, the real risk in reversing what nature is suppressing is that you'll get overgrowth, and the real risk there, I think, is cancer.
I think secreted factors are really interesting and important molecules to pursue in the regenerative medicine space. First of all, because they're biologics and they can be delivered. They can be synthesized in the laboratory, delivered, and we know how they work. And there are many secreted factors that act directly on the regenerative program. Some are generic, as you suggested, some are specific. So a generic one would be TGF-beta. Transforming growth factor beta is a secreted factor that stimulates fibrosis and collagen deposition. It's involved in wound repair. In some cases, its activities are beneficial. In some cases, they're detrimental. But there are tissue-specific secreted factors that are classic examples of regenerative molecules. The best example would be EPO, related to what John mentioned. EPO is used widely in patients to regenerate blood cells. Another one would be VEGF, a secreted factor used to generate blood vessels, and anti-VEGF therapy is widely used now in macular and disorders of the retina. So I think these types of secretive factors are very powerful. We don't yet know the identities of all of them. There will be many that are involved that interface with the immune system, and we heard a number of great talks at this meeting already about how immune cells play such an important role in regeneration. And once again, it comes back to, it's a two-sided coin. With the immune system plays a critical role in regeneration by clearing out debris so that you can rebuild the structure, but the immune system can also play a negative role in regeneration as well. So it comes back to understanding how the biology works, but secreted factors, I think, are a really important avenue for the future.
I think getting to your question about will there be commonalities amongst regenerative processes in different tissues, I'm surprised that there aren't more common cassettes or pathways that have been identified in stem cells in multiple tissues. And in adult organs and tissues, in general, most of the pathways we find seem to be tissue specific, like EPO, like VEGF. And while the same players might be used again in terms of transcription factors and intracellular transducers, there aren't that many common adult stemness pathways that have been identified, perhaps to one's surprise. So I'm not sure there's going to be so many factors, unless we just haven't found those pathways yet, that will sort of boost everything, fix all tissues equally well. And we might have to work them out one at a time.
I think another challenge is going to be how do you benchmark the human population so that you know which groups you can cluster for a particular treatment. It's already been brought up, aging is a huge variable, and how the immune system, how cells respond are not going to be identical for a teenager, a 30-year-old, or someone who's in their 70s. And understanding those molecular changes probably is going to mean that you change quite dramatically even how you approach the therapeutic modalities.
So, please, Steve.
Yeah, I was just going to ask, I can hardly remember a medical advance that was more hype than IPS cells. We went in 6 years to no.
And we're not over yet. It's still in the midst.
So that was going to be my question. Where do we stand now? What do people think now about the potential of IPS cells?
I'll just jump in on the aging part. iPS cells are great in terms of giving you certain specificity, but it doesn't mimic any age. You get a epithelial cell, you transform it and dedifferentiate it, it then goes to some unknown age. So if one is thinking about, let's say, degenerative diseases of aging, you're dealing with a neuron that's probably a child-age neuron that came from a 60-year-old. So transdifferentiation might actually be a better approach to that. It's not that you can't use these differentiated systems.
So Steve, I think you're commenting on the hype, not of the biology, but of the application, right? Because you'd agree that it was an important advance biologically. So I think it was exactly sort of as several people have mentioned here, really the notion that cells will be made, transplanted, and diseases will be cured. And that was the naive part, right? But I think what has been, for me really surprising in a way, is the use of iPS cells from patients to create models of disease for studying the diseases and for screening for therapeutics in ways that I would not have predicted, or I guess would be so successful in cellular systems being useful for modeling diseases, even complex diseases. I mean, so I would say that we're in the early stages of figuring out what the real applications will be, but it will be more than, for sure, the initial hype of, making neurons, transplanting and curing strokes, and making heart cells and curing. So that was the naive part, or, whoever was hyping that, was a combination, obviously, of the media, not just scientists. But I think there's really, there are many applications beyond that that are evolving, and the field will evolve to where that will be a standard for understanding diseases as well as treating diseases.
You just mentioned that I think there's also a real evolution in our understanding of reprogramming biology. So the initial idea was quite simplistic. You simply bashed a cell with 4 factors and it would immediately obey the signals to become completely pluripotent and utterly sort of tabula rasa. And that has turned out to be very, very naive. And indeed, most cells in a reprogrammed environment remember far too much unless you continue to bash them over their head. In which case, then they become something quite artificial, which might not be a bad thing, but it is definitely not what's normally thought of as a stem cell. And so I think, to some extent, we're really at the beginning of a long set of research questions that have to be answered about what it is that a reprogrammed cell really does and what it doesn't do. And are there shades of reprogramming that we're not aware of, where you take a cell back but not that far, so that you can actually bring it down another path, or maybe just rejuvenate that path and bring it to a less aged state. But ultimately, I think what Rick was saying, I think it was Rick who was saying that, you know, you don't know what age you're getting when you do these things. And the fact of the matter is that the opposite is true. If you take a fibroblast out of my body, it's going to be an old fuddy-duddy fibroblast. And it might very well have undergone all sorts of, you know, epigenetic modifications which you wouldn't want to really pass on, or for that matter, somatic mutation. So I do think that the source of these cells is not just an easy question to answer either.
So I'll just comment quickly and say that I think there probably will be a few cell types that will be developed from iPS cells for therapeutic purposes, perhaps limited ones, maybe with engineering components to them, cartilage, chondrocytes, things like this, maybe components of the blood. But I agree that I think using them as models of disease and as personalized therapeutic tools can be really effective, and already has been in a few cases, and I wouldn't be at all surprised to see a lot more of that. The example of patients with long QT syndrome, unresponsive to therapies, and then making cardiomyocytes from their iPS cells and testing a whole series of combinations of drugs to suppress the arrhythmia, and successfully using those to suppress the arrhythmia back in the patient, are pretty amazing examples of using iPS cells for therapies.
We have a question over there.
There's a question in the back as well.
Yeah. Why don't we go there first, and then we'll go to you. And Brian, you'll be next.
So I don't know if it's on or not, but so one of the things that I think regenerative medicine is good for all of us is exciting the public, because I think the public is really, really engaged in anti-aging and in pro-regenerative therapies. And parallel to that is exciting industry and exciting innovation and tying commercialization around some of the regenerative medicine development. So do any of you have any comments on commercialization as a partner in advancing regenerative medicine?
I don't know why they're looking at me. I'm not an authority on this, but I've thought a lot about it. I think regenerative medicine has a great future in therapeutic development, but I think we're in an era where you have to be very pragmatic with respect to developing new drugs and new therapeutics. And the way that science is done and the way drugs are developed has been segregated over the past decade or so. And the way it is done now, and it's continuing to move in this direction, is that fundamental discoveries must take place in academia, and understanding how biology works and understanding how development and regeneration and signaling networks come together, how those things take place, need to happen in academia where there's no restrictions on what you work on and you can simply try to understand knowledge for the sake of understanding knowledge, whereas the translation of that knowledge and the development of new therapeutics and drugs in the regenerative medicine space needs to take place in the biotech or the pharmaceutical world. But again, it has to be done because there's little patience in the business world for exploration and fooling around and following your nose and whatever is interesting. So there has to be really well-defined goals and a clear understanding of exactly what you're going after. So I think there are some areas of regenerative medicine that are applicable, particularly using available drugs, repurposing them perhaps in the regenerative medicine space. That those types of things I think can be commercialized. But I think the days of, hey, let's go start a biotech company and figure something out with iPS cells and maybe a drug will come out in 10 years, I think those sorts of days are over.
Please.
So I just add a couple more things. So, you know, the question you asked is about regenerative medicine and, quote, anti-aging, which is a loaded term. But, you know, therapy is directed at the aging process, let's say. Now, there's going to be an overlap there, but then they're also going to be separate. Therapies directed at aging have nothing to do with what we would consider regenerative medicine, per se. In the past 10 years, it's been difficult to get interest from venture capital in biotech and regenerative medicine. And whenever I would speak with them, their eyes would glaze over and they'd want to know, well, what device can we develop? Or, you know, it was really what can come, and these are long-term challenges. There are some venture capital companies right now that are investing more in long-term, investing in long-term projects that may have a specific regenerative medicine focus. We'll see if that's successful, and that will lead to other companies joining in if they are. In terms of kind of the aging spectrum, you know, Rick and Steve and I were talking about this today. Some of this is in regenerative medicine. Some of it is in just developing therapies that slow down tissue decline. Now, that may be regeneration in some senses. We were just talking about how the FDA seems to be very willing to now talk about this in terms of approving drugs that would have a directed primarily at prevention of age-related diseases in a general sense, of which, you know, there are now good examples in animal models. So yes, people get excited. You know, it's good to have the public informed, but at the same time, you don't want to overhype the potential and then have setbacks, and you know that can be detrimental to the field as well.
So I want to comment, just follow up briefly on what Tom is talking about related to aging. You can think about, I think often, and maybe the context of regeneration here is really to introduce, replace, or to stimulate the growth of tissue that's damaged. There could also be a version of what you might call molecular regeneration, where during aging, which is itself, of course, not a disease. Aging is a natural biological process. But obviously, the path of aging can either be towards a centenarian with great cognitive abilities or individuals who become at risk for a wide range of degenerative disease. If one could understand how to keep cells healthy, so in many ways, if you understand, let's say, the quality control mechanisms, who's there to get rid of the junk in your cells? How do you keep a balance and function? Can you stimulate a process to keep cells robust? That's an interesting process as well. But again, you have to know literally, it's what we all call molecular or precision medicine, that each person's going to be different. It's very different than what we do in our laboratories, where we have to use model systems, whether they're mice or other biological systems or flies or C. elegans, worms. We use them because we have to use organisms that are essentially genetically identical so we can get statistically significant results. And then we have to apply it to you, where everyone's different. And that's really the challenge that we're faced with. in terms of molecular profiling and understanding. So I think a lot of the concern of pharma, so Jeff Kelly and I have a pharmaceutical company in Cambridge, Massachusetts, really set around small molecules for age-associated degenerative diseases. We don't mention the word age because, again, that's a very difficult thing to want to do trials towards, but you can do trials towards cystic fibrosis. You can, in a sense, repair the damage of one mutant protein to restore the function of the lung tissue. That's a different kind of regeneration, but it does restore the function of that lung tissue back to eventually a normal state. Whether, I don't think that's going to be possible with Parkinson's and ALS if the patient already has severe neurological disease, because then you're going to need regeneration. You can't just repair what's mostly gone. But if you could detect the changes earlier, and I think that's what's missing now, is we use clinical neurological indices. I think, Tom, you're the one who can talk to this, but by the time you see a patient who thinks they have Alzheimer's disease. It's too late. But if we could have seen that individual and had some molecular evidence 20 years earlier, perhaps, then you might be able to do something.
Thank you. We had a question back there?
Yeah, I had a kind of a question going back to reprogramming, but kind of on the endogenous side of things. So in any of your model organisms or tissues of choice, in an aged environment, when you have regeneration of a tissue or a whole organ, what do you think is the age of that regenerated tissue or cells? To kind of get back to, if you do need to have a reprogramming event or some step back to regenerate a tissue that wouldn't normally occur, do we think that all of these aging processes at either the tissue, systemic, or cellular level are being maintained in this new tissue?
Most of the reprogrammed cell types that I'm aware of, maybe the other experts here will know of different ones, but most of the ones, particularly in the heart and other tissues where reprogramming has been studied extensively, are quite immature. So they come from iPS cells and they traverse through the normal developmental pathways, and so they're quite immature. And this becomes a challenge with respect to cellular transplantation therapy, particularly in an organ such as the heart, or I think the brain would also be an example of this, where the cells within that organ have to be very intimately interact with each other, and to graft immature cells derived from iPS reprogramming into that mature organ and have them establish the appropriate interactions with other cells in the heart, for example, where the heart must maintain a second-to-second contractility with no disruption, else it can create arrhythmias. I think those are real challenges. So how to generate mature adult functional cells and incorporate them into an adult organ is a challenge. Now, there will be cell types, I think, that lend themselves to reprogramming technology, and maybe those will be cells that can exert their functions more in an isolated manner. Pancreatic beta cells, for example, cells that can secrete insulin. Of course, generating those has its own set of challenges, but it may be possible to take secretory cells like that, or perhaps skin cells or liver cells, cells that don't have to be so intimately integrated into an organ. Maybe those would be among the first that could lend themselves to reprogramming technologies.
I just wanted to clarify. I guess I was asking more when you have in a, like, let's say the skeletal muscle of an older mouse, when you injure it and there is new muscle formed, do you think that there is any appreciable difference of that new muscle that's formed on like a large injury scale compared to all the surrounding muscle? We know that the nuclei are more centrally located, but would you like to comment on the maturity or maybe how much junk has been built up in those, or do they have a fresh start, that patch of new regenerated tissue?
I think what you're asking is a fundamental question that is unanswered, which is how do we determine the age of a cell? The cell divides today. Is the daughter of that cell 1 day old, or is it as old as the person in whose body that cell resides? So we at this point, we don't have a molecular definition of cellular age. So in that sense, your question is, how do we even tell? How do we tell that a cell that is dividing, is it retaining youthfulness, or is it accumulating age as every other cell in the body is accumulating age? So I think we don't know the answer. I mean, I can give you a perspective, which is cells that are not dividing continue to age. Neurons, cardiomyocytes, they change over time. Refer to that as chronological aging. Cells that divide accumulate other kinds of changes, like what we call replicative aging, which can lead to cellular senescence. So in a sense, it's difficult to look at a cell that is now dividing and say, how old is that cell? But there is both a chronological and replicative history to every cell. And in a general sense, I would say the cell is as old as the environment in which it lives, is partly what we see. We don't see, just because a cell is dividing now, it's not like a reprogrammed cell. It doesn't become young again just because it divides. So I think there is this cumulative burden from chronological and replicative aging that is carried along with all the cells in the body. So, I mean, again, it's a little bit, until we have that molecular definition of cellular age, we really can't answer that question very easily.
Bram, is there a question?
Hi, thank you. Can you hear me okay? Yeah. I've heard a number of things that really have made me curious about a particular therapeutic strategy, but really hasn't been addressed fully. So Nadia and John touched on, the hematopoietic environment, the bone marrow, and so forth. The critical nature of the immune system in regeneration. And so things like small molecules, also VEGF and EPO, and these molecules that are useful. I wonder what the panel thinks about utilizing mobilization strategies that have been used in bone marrow transplantation for a long time to generate cells like hemangiocytes that are useful in the angiogenesis for ischemic tissues and so forth. Is there some promise in those strategies, do you think?
I think it will depend. It will depend on the tissue and whether the body harbors the resident stem cell population that can replenish that tissue. So as you talked about, and as you raised the point, the blood cell system is kind of extreme end of the spectrum with respect to its regenerative power. I mean, it's just stunning to realize that a single cell can reconstitute the whole blood system. There's not that many other regenerative tissues like that. I mean, skeletal muscle, as we've talked about at the meeting, certainly has its own resident stem cell population that can be mobilized to regenerate a large portion of the musculature. And liver can regenerate half of itself probably, following an amputation of the other half. And there's the inside of the intestine. The intestinal lumen has an intrinsic stem cell population that can turn over that tissue constantly throughout life. And so I think certainly for tissues that have a stem cell population, to the extent you can mobilize and amplify those cells, that's really an exciting way to go. But I think many of the tissues that we're talking about, such as, well, the brain even has neural stem cells, although they're limited with respect to the number of neuronal cell types they can generate. So to the extent that a tissue has a stem cell population, to amplify it and understand the rules and mechanisms that control that stem cell population, I think is a really promising direction.
Just might add another angle on it, which is you may have the best cells in the world, but if the environment is producing a massive fibrotic response to an injury and in a continued inflammatory environment, it's going to be very difficult to get any response to any growth factors or any cell type. So I think we brought it up a couple of times, and my own particular current passion is the concept that we need to produce the environment in which these stem cell therapies can operate, in which the normal regenerative processes in your body can operate. And many of the problems we see medically have much more to do with the impediment and imbalance in the immune system is imposing upon the regenerative response rather than a problem with that response in its sort of pure state. So that one of the obvious things is to think about how the immune system responds to various stimuli and try to coax it back into being, as good as a zebrafish or an axolotl and staying out of the way.
So I think one of the most exciting areas about that very attractive idea of figuring out how to mobilize stem cells better than we understand now is the combination of genome editing and using stem cells to cure genetic diseases. And I actually think that may be where the biggest impact of our understanding the biology of regeneration stem cells may be seen. I think theoretically we should be able to cure many of these genetic diseases now. And I'd actually be interested in the panel's opinions about whether that's true, whether we'll see in vivo genome editing before too long, or whether it will be restricted to ex vivo. But I do think that being able to target a stem cell in somebody with a genetic disease, fix the mutation and put the stem cell back, or target it in vivo, will be a really exciting area that'll come on quickly. And if we can mobilize or amplify the stem cell population that we're trying to hit, that may be extremely important as part of that. I know Eric's been thinking about this a lot in muscular dystrophy. And in sickle cell anemia, for instance, it's more attractive to think perhaps about being able to target the hematopoietic stem cell ex vivo, if only we could grow it well, and put it back for those sort of things.
So I'll make a comment on this because I'm working on this really actively in my lab. And I think this is one of the most exciting and promising avenues in regenerative medicine with real near-term rewards. And the reason I think that is manyfold, but one reason is because for monogenic disorders that can be subjected to genome editing, you know the precise molecular lesion that's driving the disease. There's no guesswork as to what's causing the disease. So in muscular dystrophy, for example, there are single nucleotide mutations that are inherited by these boys from their mother, and that mutation causes the disease. And I think a disease like muscular dystrophy and many disorders of skeletal muscle that are caused by known mutations may be among the first to be subjected to genome editing. So for those who are less familiar with genome editing, this is a technology where you can go in and change a single letter in the entire genomic alphabet and correct that mutation. And in skeletal muscle, this method can be applied with great efficiency because of the intrinsic stem cell population that exists there. And in our own lab, we've done proof of concept studies in embryos and have been able to correct muscular dystrophy in mouse embryos with remarkable efficiency. And so, as John alluded, can that technology be adapted to adult animals? And I believe it can, and we're working to deliver gene editing components to muscle cells in muscular dystrophy to edit those cells. Now, there's been concerns raised about genome editing and whether there are off-target effects or editing at sites that one might not want to edit. We've not seen those. The original concerns about gene editing errors and treatment of disease were based on studies in tissue culture dishes where the concentration of editing components was 10 or 100 times higher than what's needed to do the editing. And also in vivo, in an animal, these sort of off-target effects have not really been seen, probably because the concentration of the editing components is lower and because we have a very efficient biochemical mechanism in our bodies to correct errors. All of us have DNA mismatch errors in our bodies every day, and they get corrected. So I think, and particularly in a tissue like probably muscle, brain, or neurons, which these cells don't normally divide anyway. And so if you could do editing in those sorts of tissues in the context of diseases, some of the concerns about cancer and other things may be mitigated. So I absolutely believe we will see gene editing done in human disease within a decade, for sure.
Well, it's actually being done now. Is this on? It's actually being done now in ex vivo, people with AIDS trials to mutate the CCR5 HIV receptor in their white cells ex vivo, putting those cells back in and showing their resistance. So there's no question that people will move it quickly to humans, as soon as the proof of concepts are there.
I think diseases like, so muscular dystrophy will be among the first monogenic disorders to be subjected to gene editing. Diseases like Huntington's disease, ALS, spinal muscular atrophy, for which we know the exact mutation and we know how to do it. I think those are all really interesting disorders to test in this context.
We have a question here from Steve.
It's been a stimulating discussion. So could the panel tell me whether or not you think there actually is a resident cardiac stem cell that can make a cardiac myocyte? And if you think there is, could you please tell me what it is? So
Steve knows he's stirring a hornet's nest.
Well, I think we know that what it's not now. I think we know what cells are not resident cardiac stem cells, but do we really think there's one there that we can activate to make new cardiac myocytes, or are we going to have to put some other type of cell, an iPS type cell in the heart to make a new cardiac myocyte?
I'll be happy to give it a shot. My own opinion is that despite 10 or 20 years of people aggressively looking very hard for the resident cardiac stem cell, I'm not convinced that any of the evidence suggests that it exists. So, I'd love to see someone find it. I'm not ruling it out, but I'm not yet convinced that there is one. And I do think there's a small amount of regenerative potential in the heart. I still think that's actually an open question. I don't think it's proven, but I think the evidence suggests there probably is, and that it likely comes from pre-existing stem cells. But it raises a really interesting question, which is that despite the fact that we know that many organs and tissues can regenerate, it's amazing how little we know about how they regenerate. So even the liver, great debate still exists about what the cell is, what the process is. In the kidney, which we all know makes new tubules frequently, we don't know where the cells come from. And in the gut, there's still this debate about exactly which cells are contributing to the turnover of the lining of the gut every 2 days. So, it's pretty remarkable how little we know about regeneration of adult organs.
I think we do know quite a bit about the heart. It's just that we don't want to really hear what people are saying, which is it's a reactivation of the proliferative cell cycle in a cardiomyocyte, and if that doesn't happen, well, it happens very well in fish, it happens very well in salamanders, and it happens probably very, very well in neonatal mice. But that's not sexy. That's where differentiation is sort of dropped off the face of the earth, and the fact that you would de-differentiate a cell, we're not even sure what that actually means, and yet that doesn't appear to be the focus of the majority of people who are asking this question.
I want to sort of pivot a little bit based on what everybody's saying, and that is the NIH is sort of renewing this push to translational research. And I think some of the comments that have been outlined here are some of the dangers behind that. Obviously, science is an ever-changing discipline. We're getting new knowledge every day. And as leaders of your respective fields, do you feel that push from the NIH to translate your research? And if you do, how do you handle that?
I'm not here yet. I'm not looking forward to it.
Well, I'll say that it's been an active point of discussion at the NIH recently about the scientific population's response and to that. I sit on the council at Heart, Lung, and Blood, and it's notable that most of the calls for applications, the special RFAs, have to do with translation. And when I have frequently pointed out that it's discouraging for the basic scientists never to hear a call for basic science, they pull up all the numbers, which suggest that there hasn't been a real drop in the funding for basic science as part of their portfolio. but that they need to encourage more and more translation. There is still funding for basic science. It is still a major part of the NIH portfolio. But Congress pushes hard, the public pushes hard, NIH responds to those calls and encourages the translational components. There was a very interesting letter from the head of the review agency at the NIH to all chairs of study sections, saying that the lack of a translational component to a grant could not be used as a criticism of the grant. And if it came up in discussion, the chair of the study section should say, that's not an appropriate criticism of the grant, in response to this sort of worry that everything has to be translational. So those are my thoughts.
In Australia, the lack of a translational aspect to the grant is a sure sign of the grant will not get funded. So it's it's become extraordinarily polarized towards translation. The problem is that if you do the statistics, the number of actual breakthroughs in medicine that have come from directed research that was answering some call for a translational application are minuscule. Anybody who comes up with a really obvious answer to where a cardiac stem cell comes from is going to sit back and go, oh, that's interesting. Maybe I'll look for a cardiac stem cell in a bird. No, they're going to say, let's go to the clinic. I mean, it's not that difficult to figure out where I think we're all sensitive to the idea that our research should have applications when possible to human health. But this concept that by having some top-down mandate that we're all going to translate is going to wake us up to the wonders of our science being relevant to human disease is just ludicrous.
I worry that some of this push is driven by the pharmaceuticals industry of defaulting on basic science and thinking, as was brought up already, that it's the responsibility of the academic researchers to do all their discovery and that they're going to be the clever ones to take our discoveries into products and treatments. And so the consequence is that we're feeling the push, I think, inappropriately because the pharmaceutical industry has, for example, in the areas of neuroscience, major pharma has shut down neuroscience departments. They've just said, we're not going to study the neurodegenerative diseases. Too complicated. Well, it's not that. They're actually shifting it back to academic laboratories. And so I think we have to be cautious that our responsibility is to make discoveries, to understand how things work. And I think what you've heard here is even great discoveries, well, stem cells first and then the iPS cells, we don't understand enough yet to be able to understand what circumstance is the right one to apply. And the thing you have to worry about is you do it prematurely, and there's going to be a cost to a lot of failure. I mean, pharmaceutical industry is not willing to take risks. They are the most risk-averse unit. So I think we're caught in a very difficult position now, and I think it's really important for basic scientists to continue to argue strongly, in a sense, push back for the importance of discovery. And when discovery is ready, yes, people will be there in the academy, in biotech, in pharma, to take the great ideas. But to assume that we're going to be the ones to do the translation, I think that's not what we're good at. We're good at following our nose and finding interesting things. Pharma doesn't do that. Pharma works on a tranche system that says you must do this by this time because that's how the payments are made. It's a very different model.
I will just add that I think that the public's call for translation is completely understandable. And, it makes sense if you've got family members who are ill to want somebody to come up with a cure for the illness. And it's rational to think that scientists should be spending their time figuring out how to cure the disease, not studying those flies and worms, But it comes from a lack of understanding of how great discoveries have actually been made and how great advances have actually been made. And so I think that it's academia's job to hold the line because we think we know how great discoveries are actually made. And we should do our best to try to teach the public how great discoveries occur. And I think it begins in schools, and that you have to get to kids young about how science works, tell the stories of how great medicines were made. And I don't think we do that very effectively in this country. I will say I thought the show, The Emperor of All Maladies, I don't know how many people saw that, about cancer research that was on public TV recently, was a terrific example of that, giving the stories of basic research, not particularly directed, coming up with the best cancer therapies.
You guys want to add anything? Oh, David.
Just a comment. Thank you. I would hope that we would not separate ourselves from the bioethical dilemmas that come up from the pressures of translational research. There are an awful lot of medical people out there who are absolute charlatans who are willing to take the latest discoveries like stem cells, et cetera, and tell patients, I can cure your ALS or your Parkinson's disease, your heart disease, whatever. And they seem to be able to do this with absolute impunity. And so I think that all of us should take a stand against this and make sure that we incorporate that into our profession. Hear, hear.
I think, in a discipline like regenerative medicine, one could easily see the potential of this type of research and the application for human health. And presumably this is going to impact students at all levels as well. And we see an increasing number of students who want to take their basic research to a translational perspective. And as mentors, do you find that it's a difficult balance between encouraging the discovery of the basic research, tempering with the expectations of what that finding could mean in a translational setting in your own respective labs? What about you, Tom, or Eric?
Well, I think students sort themselves out based on their interests. And I think as a mentor, if you provide an environment that values hardcore science, rigor, lack of hype, that's the platform upon which students can train, and some students gravitate towards basic science for the reasons that we've talked about in my own case, and some gravitate more toward the translational, some gravitate toward teaching or government. There's many different career opportunities obviously in this business. So although I'm a dedicated academic, I don't think academia is the only respectable career path in science. And so I think translational research, drug development, animal model development can be equally as valuable as basic science. So I think the responsibility of a mentor is to provide the trainee with an understanding of all the opportunities, the positives and negatives, and let them make their own decision based on their risk tolerance and other personality traits.
Do you have a question back there?
In research. And Dr. Nadia, right? You mentioned about the environment. So have you heard about these mice that they did they connect a young mice with an old mice, they put the blood into the old one, and oh, so you are the guilty one. Okay. So my point is, do you think instead of working with stem cells, like the cells as such, you should work more with the environment surrounding after damage?
Well, that's what Nadia was saying, and I completely second that, which is one can't view the stem cell biology independent of the environment in which they live. And I mean, one can study them independently, but in any kind of application, one has to be looking at both. And it's a little bit easier to imagine manipulating the stem cell as opposed to manipulating the environment, because it's just much more complicated. But that's, those experiments are really addressing that question of can you manipulate the environment and can that then provide a better environment for stem cell engraftment? The answer seemed to be yes. So I think if you're asking, is that the approach? And I think most people would agree that the environment is becoming increasingly recognized as a critical factor for any kind of stem cell function, stem cell therapeutic.
So if there aren't any other questions, I want to end with one question that I'd love to get everybody's feedback on. So obviously, in addition to having this conversation with scientists in the audience, there are also lay people who are hoping for the next great discovery, both here as well as online. So I thought maybe we can close by, we started out this conversation with this debate of hype versus promise. And I thought maybe each of you can just provide a few last words towards that regard to the more general audience. So we'll start with you, John.
You want hype or promise?
It's up to you.
I think one of the almost exciting things about science is that it's so unpredictable. And I have no doubt that there'll be advances in the regenerative therapies and there'll be cures of genetic diseases and that sort of thing. But predicting when they'll happen, I think, is a fool's game. And advances happen in quantum leaps, in unpredictable ways. And all I can, suggest is that if we don't work hard, if we're not, don't have resources to do the hard work, those advances won't occur. But trying to predict when they'll occur, how quickly, I don't see a great benefit in doing that. And frankly, I don't think the public craves for that. I think they want to see advances. They want to know that the work is being done with integrity. And in general, I think that they trust the population of scientists and physicians to do their best towards those ends. And they want to know that there's real integrity at the other end of the dollars that they're sending our ways. So I hope that there is.
The public allows us to do what we do. It's really a remarkable process where it's because of the public support we get to do science and we get to make discoveries. So I think it's very important for those who, in any public sector, to go out and talk to others about what is it that scientists do and what's the process. Most people don't know. They just think, well, we're going to cure cancer tonight. Or didn't we cure cancer yesterday? It's not so simple. And I think what we've talked about is that it's a long process. It's discoveries. It's sometimes random walks and people putting together observations. But it's not that we're asking anyone to be patient. It's actually the process of human inquiry and discovery. It's very hard. We're not engineers. An engineer can look at a bay and say, I'm going to build a bridge over that bay. They know how much steel they need. They know what all of the calculus and geometry that's required to do it. We're looking at the complexity of life and trying to understand one or two things in that complexity and to know how we can talk to that cell, that tissue, in all of that complexity. I actually think it's kind of amazing what we do as biologists. And it's remarkable that the progress that we can make because of the diversity of organ systems and tissue systems and model systems, and actually the diversity of scientists who come to bear. That we're not all trained the same, we come from very different backgrounds, and that's the uniqueness of our discovery process.
I don't have a lot to add. I guess I would say that being a scientist is an incredible privilege. Society enables us to do whatever we want to do and to make our own judgments. Being a scientist is like being an artist, creating, understanding, looking at the world and understanding how it works and creating a body of knowledge. Scientists typically are not trained or prepared to deal with the public and to deal with the lay press, and they oftentimes have a tendency to promise more than they can deliver, because they want to get funding, or they want to tell a good story, or they want to respond in a positive way to questions that are being asked to them by the lay press. And I just think it's important to dial hype out of this business and let scientists do their jobs. And at the end of the day, I think great advances will come from that.
I don't have much to add either. My first thought was the comments that have been already stated, and that is that when you raise this issue of hype and hope, the word that comes to mind is education. Because education reduces the hype, but doesn't diminish the hope. And I think, as Eric was just saying, we're not trained very well during our graduate years, postdoctoral years, and professorship years to educate the public or educate the politicians. And we do a little bit of that, but clearly, as a field, we don't do that well. As a profession, we don't do that well. And so I think the burden is upon us as scientists to maintain hope based on clear past successes, but not predicting what the next one will be, just like John said. But that kind of education can temper the hype that appears in the media. We need more scientists speaking up instead of the media speaking up about these things, and then providing that background. It's not saying there's no hope. There's still plenty of hope. It just has to be temporary. And I think education of the public and in particular politicians is a major step that will benefit the whole profession, the whole country.
One of the changes and advances that I'm looking forward to the most in the years I've got left as a scientist is to see what's going to happen when the public becomes really engaged in science. And I think that's a lot sooner than we have ever seen before due to the advent of genome sequencing. So within the next decade, people are going to actually request for their genomes to be sequenced. They're going to see what that actually will do for their own medical treatments and prognosis. And it will also start to contribute to a vast body of knowledge that we as scientists can't gain on our own. We need the cooperation of the public to do this. And so with 100,000 genomes and the associated medical information that will start to pile up in the next decade, we're going to be able to see much more clearly how people work, how they differ, and how science can help them with their individual health issues. I feel extraordinarily optimistic about what we're going to learn. I know that there are ethical problems with all of this, but in countries with national health services like the UK or Australia, a lot of these questions have just been completely bypassed, and the work is continuing. So how is that going to actually impact on regenerative medicine? Well, we've talked about the accumulation of mutation and aging. We've talked about the changes that we see in our own bodies as we age, but we've also seen that people do differently after they get injured or suffer a degenerative disease. And that's where I think the real progress is going to get made faster than anything else. It may not be a basic change in the way we treat people on a grand scale, but it might actually be how we dial up and down medicines that will make huge differences in the way people respond to treatments. And so I think that what we need to do in terms of education is to tell the public how exciting it's going to be for them to be part of this experiment rather than just watching it from the sidelines.
Thank you. So if there are no more questions, I really want you guys to join me in thanking the panel again for a really great discussion. And they will be around for more conversations. Thanks, Tom. Thank you. Thanks, Eric. Thanks, Tom. Thanks, Nadia. Thank you so much, guys.